Good day, everyone. I'm Angel from Group 1, and today we'll be discussing microbial technology based on Chapter 5 of Biotechnology Fundamentals by Fergus Alam Khan. For transparency, AA tools were used to assist in organizing and refining the content of this presentation. Now, we'll begin our exploration of the microscopic world by first defining what microbes are. Microbes are single celled organisms, and they represent the earliest and most enduring form of life on Earth. Fossil evidence show that microbes existed more than 3.5 billion years ago, long before plants, animals, or even the dinosaurs appeared. For about 2 billion of those years, all living things were microscopic, mainly bacteria and archaea. These early microorganisms shaped our atmosphere, soils, and ecosystems, laying the foundation for complex life. Even today, microbes remain essential to every biological process on the planet. They are literally everywhere, on our hands, in the air we breathe, in the food we eat, and inside our own bodies. In fact, there are more microbes on a person's hand than there are people on Earth. Without them, we cannot digest food, plants could not grow, and organic matter would never decay. Microbes recycle nutrients, produce oxygen, and maintain global balance. They are quite literally the unseen engines of life. Well, our understanding of microbes began with Anthony van Leeuwenhoek, who first observed bacteria in 1676 using a single lens microscope of his own design. He called these creatures animalcules. And then, a century later, Louis Pasteur demonstrated that fermentation was not a chemical accident, but the result of microbial activity, which disproves the old belief in spontaneous generation. Next, Robert Koch, together with the help of Pasteur, they established the drip theory of disease, which linked microorganisms to infection. Koch's postulate remained a cornerstone of microbiology, and his work on tuberculosis earned him the Nobel Prize in 1905. Later, in 1977, Carl Woese used 16S rRNA sequencing to reveal that archaea represent a separate evolutionary domain distinct from bacteria, which transformed how we classify life. Now we move to the anatomy of microbes. Bacteria exhibit remarkable diversity in shape and size, known as morphology. They can be spherical cocci, round-shaped bacilli, curved vibrio, spiral spiralia, or tightly called sporochetes. The shape affects how bacteria move, attach to surfaces, and acquire nutrients. Some live as single cells, while others form colonies or chains. Many bacteria also develop biofilms. These are dense communities attached to surfaces which enhance survival and antibiotic resistance. Inside the cell, as shown in the figure, bacteria lack membrane-bound organelles. Instead, their plasma membrane encloses cytoplasm and controls molecular exchange. Bacterial ribosomes synthesize proteins, and some species contain specialized microcompartments, such as carboxysomes, which concentrate key enzymes. In photosynthetic bacteria, the membrane may fold extensively to form light-harvesting structures for capturing solar energy. Externally, most bacteria possess a cell walls made of peptidoglycan, which maintains shape and prevents rupture. We'll distinguish two main types. First, the gram-positive bacteria with thick peptidoglycan layers and teichoic acids, and then the gram-negative bacteria with thinner walls surrounded by an outer membrane rich in lipopolysaccharides. The figure shows these key structural differences. These variations also determine how bacteria respond to antibiotics, like penicillin and vancomycin, for instance, are effective mainly against gram-positive species. Many bacteria also possess additional surface structures as layers that act as protective coats, capsules that aid immune invasion, and appendages such as flagella, fibrae, and pili. Now that we have discussed the anatomy of microbes, we have a clear view of how their structure supports their survival and function. This understanding will serve as a crucial foundation for our next topics. Thank you.